My name is Adriana Ruiz-Rizzo, and my talk is titled Visual Processing Speed is linked to functional connectivity between right frontoparietal and visual networks. Visual processing speed is a relevant visual attention function that reflects attentional capacity or how fast we can process visual information. It is relevant for daily living activities that rely on visual information, like making decisions, encoding information, or for spatial navigation. We can measure visual processing speed in the lab by using a whole report task, where we present participants with arrays of letters very briefly, and we ask them to report all the letters they are fairly certain they have seen. This way, we measure accuracy instead of motor speed. What we do instead is vary in exposure durations, so that we can estimate visual processing speed with an exponential growth function where by increasing the exposure duration, the mean accuracy of report also increases. The slope of the curve at its origin represents parameter c, or visual processing speed, and is quantified by the number of letters per second. If we want to assess the neural correlates of visual processing speed, we can turn to functional connectivity, which can be obtained from resting state functional magnetic resonance imaging, and more specifically from the spontaneous fluctuations in the blood oxygenated level dependent signal, or BOLD signal. By using varied methods like independent component analysis, we can obtain different sets of brain regions that reflect brain networks. By using these approaches, previous studies have shown that the connectivity within the singulo-percular network and the connectivity between the singulo-percular and the right frontoparietal network is relevant for visual processing speed. However, from a theoretical perspective, visual coding regions should also be involved. And thus, in this study, we investigated whether these higher order visual processing speed relevant networks also connect to visual regions to support visual processing speed. Therefore, we assessed a sample of 48 healthy participants who underwent resting state functional magnetic resonance imaging. And for the analysis, we used the entire frequency range available in our data by separating into frequency bins from the slowest, or slow 5, to the fastest, or slow 2. We analyzed the between network functional connectivity between the singulo-percular, the right frontoparietal, and the visual networks. And then we assessed the association of this between network connectivity with visual processing speed. We identified all the networks, as I said, using ICA across frequency bins. Here you can see the between network connectivity between the networks of interest. And as you can see, from the slowest to the fastest, it was mostly negative, the connectivity of this higher order with visual networks. In particular, for the singulo-percular network, it was strongest in slow 4 and negative, and also in slow 3 and positive, and more specifically with the insula. As for the right frontoparietal network and visual networks connectivity, we saw that it was strongest and negative in the slowest or typical range of frequency bins. Next, we examined the association with visual processing speed. And we did that by analyzing 5 linear regression models, one for each visual network identified. And as predictors, we used the connectivity of these visual networks with the singulo-percular and the right frontoparietal networks. So here I'm showing the estimates and the 95% confidence interval of each of the predictors related to the singulo-percular network. And as you can see, none of them significantly predicted visual processing speed. In contrast, when we look at the parameters predictors involved in the right frontoparietal network, we see that for 2 models involving 2 different visual networks, there were 2 predictors, slow 5 and slow 2, that significantly predicted visual processing speed. So in summary, we found that functional connectivity with visual networks and the singulo-percular network was strongest in slow 4 and slow 3, the second fastest frequency bin. For the right frontoparietal network, it was strongest and negative in slow 5 and slow 4, the typical frequency range for analyzing resting state fMRI data. Most importantly, the right frontoparietal, but not the singulo-percular network connectivity with visual networks was associated with visual processing speed. And this was observed in slow 5, but also in the fastest frequency bin of our data. These results suggest, in light of previous evidence, a functional hierarchy involving these 3 networks where the right frontoparietal could be the interface between the cingulate, ocular, and visual network.